Welcome everyone to Biology 380, Food and Industrial Microbiology. So this is going to be a guest lecture. So Dr. Tabatabaee invited me to come here and give a little lecture, and I'm going to try to aim for 40 to 45 minutes on the specific topic of industrial biocatalysis. This is a kind of broad-ranging topic, and I'm going to try to give you an overview of what it can do and what potentially it's going to be useful for in the future. So this is not going to be about food directly. It is definitely going to be about the industrial aspect, and it's going to use microbiology, but I'm not going to cover a lot of the types of microbes or the types of either yeast or bacteria that are being used. I'm going to focus more on the applications, and my goal here is to give you an overview of what's out there and what the process is going to be like for, I don't want to say the future, but it's definitely a trend now that will change the way a lot of things are created. Mostly chemicals, a lot of pharmaceuticals, sometimes also biofuels. So that's a way to get out of the oil industry, which, as you probably know from the geopolitical events that are happening these days, are not always the best resources to be depended on when they can only be found at one place on the planet. So industrial biocatalysis, if it creates biofuels, can give you, well, you can create your plant, put your plant anywhere, and you're going to get your resources out of it. Maybe it's going to make more sense when I get deeper into the discussion. So allow me to introduce myself. Probably people here might not be familiar with me, so why should you listen to me about this topic? I'm a researcher here in the Department of Chemistry at UBC, and I'm an organic chemist by training. But before my time at UBC, I worked at ASI Research Institute, which is a pharmaceutical company in Boston in the US. And later on, I worked at Pfizer, obviously a pharmaceutical company, in Groton, Connecticut. So I worked specifically at the largest site. It's called Global Research and Development there. So there's 12,000, this is a small city where there's 12,000 scientists who are focusing on developing new drugs, there's not much vaccine happening there. This is not where most of the bulk is. And there's probably 5,000 chemists and about at the height, probably 8,000 biologists there who are responsible to screen drugs for diseases, find new metabolic pathways, try to define better ways to treat diseases. So I'm showing two molecules here. I'm a chemist, so I'm not going to talk much about the molecules per se, but we have to acknowledge the fact that they are structures. So these are mostly visual supports. They're not going to be, I'm assuming you will not get tested on any of that stuff, but I want you to be aware of this. So at ASI, I worked in the Department of Process Chemistry on eribulin, which is one of the most potent anti-cancer molecules that exist out there. And at Pfizer, I worked in the Department of Process Chemistry, and it's going to come into play a bit later on, where I worked on a different project where the goal was to create some molecules with chiral centers on it. So does it matter? No. So it's not about organic chemistry. This is about the fact that these two companies, and now more and more companies, are using different unconventional techniques that don't rely on classical organic chemistry to create compounds. And there's two reasons why people are using biocatalysis instead of classical chemistry. Is first to reduce waste, because the types of reagents that are being used are not the same at all as chemistry, and for sustainability, because once you get the proper bacteria or proper yeast to do the transformation you want, you can keep it going. So in terms of if you've ever done a synthesis, you start with a starting material, and the more you transform it, the more you lose because you generate impurities. With biology, it's often the opposite. You can start with a certain, technically only one bacteria, and you just feed it. It's going to divide. So over time, you actually end up creating more of, I guess, your little microscopic industry, as opposed to, as long as you can feed it the appropriate amount or type of nutrient, then it will keep going. So it's more sustainable. And maybe one aspect I should touch upon right now is the idea behind chemical waste is you generate a lot of solvents when you do reactions on large scale, because you produce chemicals, polymers, drugs for the planet. And all that chemical waste is organic. Often they're halogenated solvents. It can be carcinogenic. So we try to minimize it in industry, but the industrial biocatalysis is a way around that, because the solvent is water. So as long as you control the pH of your water and you don't have too much extra components, then the waste that you're generating on large scale is going to be less damaging to the environment. Okay, so let's dive back into the topic of the day. So for what you've probably seen so far, in the class, is the roles of microbes in terms of manufacturing and in and around solid foods. There's probably a small aspect, or hopefully not too small aspect, that relied on industrial processes. And this is where my topic today fits in. So I'm going to focus on enzymatic technology, which is, can be called also biocatalysis. So let's address a few definitions first. So what is biocatalysis? So for the purpose of this discussion, we're going to limit this to a chemical reaction that will be catalyzed by a biological reagent. Most of the time, that will be an enzyme. So as many of you have probably seen in biochemistry, if you have an enzyme, so this is the cartoon version of this that is not rigorous at all, but it shows what the essential components. So you'll have an enzyme. An enzyme is a protein that is found in living systems, often a cell. And that enzyme will be able to recognize a substrate. If your substrate comes in and binds to the pocket, which is your active site where the chemical reaction is going to take place, then once it's fully seated in its binding pocket, the enzyme can do a reaction on it. This particular example is showing a cleavage. So the substrate is being cut into two pieces. So the active site will come in, cut one pieces of the substrate, and as you have severed the connection between the two, the small and the big piece, then the two components will be released from the active site, and they will be called products. So you have your substrate that's coming in, the enzyme binds it, does the reaction, and in the end, ejects the product. I'm going to address very quickly the arrows here. So the fact that these arrows are going in this direction means that this is the direction that is favored. Most of the time, those enzymatic transformations are reversible. The driving force that keeps it going towards the product is the fact that the substrate will have a better affinity for the binding pocket than the products. So there's more affinity, or there's a higher probability that the substrate is going to bind here than the products. Therefore, that's the driving force. So the energy of the products binding to the active site is lower than the substrate, so they get ejected over time. So this is like the overall transformation that happens with enzymes. So another aspect is biocatalysis. So as you probably know, the enzyme is the catalyst in this whole transformation. So the substrate will not spontaneously decompose into the two products. You need the enzyme to come in and do it. And catalysis means that it's going to not only favor the reaction, but it's going to accelerate it. So the active site is designed to actually favor and make the reaction happen very fast. So that means in your cell, you're going to have hundreds and thousands of reactions that take place at every second. Okay, so just in terms, I'm going to acknowledge the references here. A lot of the material or the ideas that I'm talking about, you can find in this Nature Review Methods primers that was published last year. You don't need to read this. It's just if you're interested in the topic, this is a good place to start your deeper readings. Okay, so now that we've addressed what biocatalysis is, I guess I didn't address the bio portion. It's probably self-evident. The bio is that it comes from a living organism. So a classical example of a enzymatic transformation is usually the transformation of starch into glucose. So in bread, in potatoes, in rice, the main white stuff that you see in there is starch. So starch is a polymer, so that means you have a very long string of repeating units that are associated together, which, as probably many of you know, is a glucose subunit. So you have one glucose subunit attached to another one, to another one, to another one, to another one, and this creates a polymer that becomes insoluble in water. If it's insoluble, then it precipitates out. Or another way to look at this is you can now have something solid, which is, you know, the bread, the white inside your potato, or the rice. So in your saliva, you have your amylase. Well, you have tons of enzymes, but one of them is the amylase. So your enzyme A's means it's going to cut. So the amylose or glucose subunits here. So why do you need this? Well, we cannot digest a polymer. Our body needs glucose to function. So the function of this enzyme is to just come in and cut all the different pieces of the starch to create, not to create, but to generate, to release the single units of glucose that now we can use as energy. I realize this is outside the window, so just move this a little over here. Okay. So you're releasing the glucose through an enzymatic reaction. So this happens in your body all the time. What does it have to do with biocatalysis or industrial component? Well, industrial biocatalysis is basically the same thing. You're using an enzyme that will do a chemical reaction. The difference is instead of being on a small scale, like in a bacteria, or in a small scale in your saliva, in your mouth, then it's going to be on large scale. So that means you have this scientist here that has reactors that are full of media that has floating either bacteria, potentially yeast, and it could also be isolated enzymes like outside the cell, but that's less, that is less common nowadays. Why is it less common? If you isolate the enzyme out of the bacteria, then it cannot replicate. You have only one reagent, one protein that's doing its job over and over, but eventually it will degrade because of oxygen, because of oxidation, because of degradation or unfolding of the enzyme. So it's not going to be active as much anymore. It's going to lose activity. The advantage of using microbes like yeast, bacteria, or algae is the enzyme is actually in the cell, and the cell machinery will first keep making more of new enzymes if it's needed, and is going to degrade and eliminate the ones that are not functioning anymore, the ones that are in excess, And more importantly, the cells are going to divide over time, so which ensures a steady state concentration of your cells. Okay, so that's kind of the overview here. So you'll have typically a warehouse with big incubators. You can call them, if they were chemistry, if that was a chemistry lab, it would be called a reactor. Often when you deal with biocatalysis, you call it an incubator. Why? Because you incubate your microbes in there. Okay, so let's address a few. This is going to be the slide that has the most text on it, so I'm going to take you through each step because again, this comes from the paper I just mentioned, and I think it summarizes a lot of the current state of the field. So biocatalysis has become an important aspect of modern organic synthesis. So I know you guys are microbiologists, you don't want to hear about organic synthesis. The thing is, all molecules of life are organic molecules. And if you don't have a way to synthesize them, to create them, then, well, we don't have them. So it's an important aspect of synthesis now in the chemical and pharmaceutical industry. So why is it successful? It's successful largely because of the rapid expansion of chemical reactions accessible. So what does that mean? I'll come back to this. That's been made possible by the tools of enzyme discovery coupled with high throughput screening or evolution techniques. Well, there's a lot of things that I need to go over here. So I'm going to talk about this in the reverse order. So what we have now in terms of sequencing and genetic manipulation tools is it's possible to rapidly evolve bacteria, or I'm just going to say microbes from this point on, because that encompasses the whole classes of cells, single cell. Yeah, let's go just single cell organisms. We have now, through phage display, which I'm not going to talk about here, a way to screen at high throughput. So that is getting mutations to happen in cells for the gene that encodes for your enzyme in a very rapid manner. We can create several thousands of mutations every day. And we have good methods to be able to test whether the chemical transformation that we want is happening or not. So what is enzyme discovery? Well, discovery of enzyme is basically looking at the genes, genetic sequences of different proteins that are in different organisms. And the databases are very powerful, and we can screen. If you find a new gene, a new potential enzyme, you screen against all the database of other organisms, and you can tell whether it resembles something that already exists. For example, if I don't know, I'm going to talk about amylase again because that's the one I just talked about. So if you're isolating a gene from a given bacteria, you can screen against the whole database that exists. And then if you find a hit, there's a lot of similarity for one particular class of enzymes. And let's say they're all amylases, there's a pretty good chance that the sequence, so that is all the amino acids that create your enzymatic protein. If you have all the amino acids that line up with a lot of different amylases, there's a good chance that your gene will be responsible for amylase activity. So why do we care? Well, the more options we have in terms of enzymes that exist now and that we have inventoried, the more options for different chemical transformations we have. Now, I'm not sure, trying to remember if I'm going to cover this or not. I think I will, yes. So we're going to cover a little bit more what evolutions mean, but the point is you can modify the amino acids in your enzyme so that it can do a transformation that may not be natural in the living system. So that means that it leads to now this is the expansion of chemical reactions. You have now new potential reactions. So this particular example here is is an enzyme that normally would take proline, which is this amino acid, and would introduce an alcohol at this position, so an OH group. So this is an enzyme that does a hydroxylation. This is an oxidative enzyme, so it adds an oxygen to it. So why does it do it? Well, because the organisms, I think this comes from plants. So in these particular plants, they need to use a functionalized proline that contains an alcohol to do something else. Well, chemists and, well, I guess I should say biochemists have managed to modify genetically different amino acids at the binding site. So instead of adding an alcohol, it's adding a chloride at this particular position. Now, this is very rare to see enzymes in nature that will introduce a halogen on a molecule. Why? Because halogens are usually bad. And so therefore, nature has not evolved to introduce halogens that would be deleterious to them. However, as a chemist, maybe we have a reason to use this. So by genetically engineering the enzyme, you can make it do a reaction now that was not possible, that is different from nature. So the convergence of fast sequencing, genetic modifications that can be tested very rapidly, and molecular modeling that allows us to model the binding sites of a given enzyme have converged to open this whole field that was not possible before. So the field was possible before, but we didn't have enough information or tools to be able to take advantage of it. Over the past 10 years, this whole field has exploded. And again, probably more related to sequencing technologies. Okay, so what's the outcome of this? It means that now we can have tailor-made enzymes. And once you have the enzymes, you can either produce and isolate the enzymes and get the reactions to happen on gram scale to kilogram scale through genetic modifications. So whether you isolate the enzyme and have it do the job for you, or whether you keep the enzyme in the microbe and get the microbe to secrete the product that you want. Okay, let's move on. So industrial biocatalysis is now used in the pharmaceutical industry all the time. So I'm just showing you here another example where the actual enzyme is isolated. So this would be, you don't have to know all these names, but this is the crystal structure of a protein that is a dimer, and it's a transaminase. So its job is to transfer amine groups. And what this is showing is that you can use only 3% weight. So that means you can only use however much of substrate you want to transform. You only need to use 3% of your enzyme, hence catalysis, to transform, to get the reaction to work. And the solvent here is water. So this is biologically produced and isolated. So it means in terms of waste, what do you have at the end? You only have amino acids and water. So the waste is environmentally friendly. So there will not be anything here that is unknown to nature. Water is clearly present in nature, and amino acids and enzymes are clearly present in nature. Will they keep destroying things or doing a transformation? Probably not, because they won't have the proper substrate, or they won't have the proper amine component needed for the reaction, which I'm not showing here. So this is an illustration from an actual drug called, actually there's a typo, I think it's stiagliptin. It's an anti-diabetic, it's a drug against diabetes. What it is now the chemists put all of this molecule together, and the last step of the reaction is to convert this ketone into the amine. And instead of using normally 2 steps using chemical reagents, they simply take the enzyme that converts the ketone into the amine very effectively. So this is one example where the enzyme has been isolated. The enzyme is in the reactor and everything is flowed into it. So you have all the pumps that are injecting the water, that are regulating the pressure, regulating the temperature to make sure that you have conditions that mimic the biological pH, the biological temperature. Biological pH is 7.4, temperature is 37 degrees. And then at the end, what you get is basically your desired product that's floating in a solution. So I'm not going to go into more details in terms of chemistry. The question I want to ask is why? Okay, so it's one thing that you can get enzymes to do a chemical reaction, but why is the world interested in this? So I kind of addressed the fact that environmentally friendliness is one aspect. So, but that's not all, you know. I guess one way to describe environmental friendliness is green chemistry. So you're now doing chemistry that's happening that is green, that does not produces a lot of waste. But the real reason why people are interested in anything is always because either you can get something that has, well, it's always because you can create something that has high value. So that is drugs and commodity chemicals. So you can get materials like polymers, you can get cosmetics like fragrances. I don't know, I'm just going to say creams here as a general term. Nutritionals, so nutritional supplements like vitamins, all of these. So instead of extracting materials from nature, or extracting nutritionals or cosmetics from animals or nutritionals from plants, maybe you can actually get them to create it from scratch, from something more simple. And why I'm actually going to spend a lot of time to talk about drugs today is because that's where a lot of the most of the work over the past 10 years has been made. Why? Because money. So it means if there's an incentive for money, then you can actually invest the time and effort and resources needed to do all the research that is needed to isolate, to genetically modify, and sequence your enzymes. So I would argue that the pharmaceutical industry has done the most to advance the field, but now all the outcomes of that research is being transposed more and more into, I would say, I don't want to say societally more meaningful material or components, because drugs are still important, but let's just say we can move beyond just drugs. Okay, so what is the process here? The process is, if you're interested in a given molecule, so this is Vindoline, an anti-cancer agent. This is Thebaine, which is this is basically a morphine derivative. So I'm not going to go through all of these molecules. You don't need to know these. The point is, you have complex organic molecules that usually come from nature. So medicine, or a lot of our tools for humans, when it comes to things we eat or put on our wounds or try to use as fertilizer, will often come from nature. Nature is our major source of inspiration. So what we have done typically in the lab is look at the compounds that have an effect, and then we try to synthesize them from scratch. But what biocatalysis is doing is now, what if we could actually, now that we know these molecules come from the plants, can we not go into the plants in the first place and see how the plants do it in the first place, and which would be through enzymes. If you discover which enzymes are responsible for synthesizing these molecules in the first place, maybe you can get the enzymes, take them out to the plant, put them in a controlled system. Why do you want a controlled system? Well, because you can get to large scale much faster. So if you want to get to large scale for, like, let's say if you need this particular a compound from this particular plant, and if you want to have enough plants to not feed, but to provide enough compounds for the planet, enough drugs for the planet. An example is latex. Latex is a natural material that comes from the latex tree. But if you bleed, because you have to cut the tree and to collect the latex, if you bleed all the trees on the planet, then after you've bled the trees, you don't have, or maybe one way to think about this is, the trees don't grow as fast, or the plants don't grow as fast as the extraction we need. So by controlling the system, we can actually control the rate of reproduction or the rate of activity of our, let's say, enzymes, so that they can produce the material that we want in a controlled environment where we don't need half a country full of jungle or forests or fields. So we can have a more efficient, therefore less costly and less environmentally impactful method. So this is kind of the whole idea behind this. See how nature does it, sequence and reproduce the enzymes that are active, and hopefully get these enzymes to act in concert to recreate the high value compound that we want. Okay, so I thought the best way to exemplify this, the whole process, was to talk about drug synthesis. Why? Well, because as I said, this is where a lot of the work has been done, and that's where you also have a lot of more spectacular examples at this stage. Okay, so before I dive into this, I think I need to introduce a little separation of two fields in the pharmaceutical industry. So when I said I was at Pfizer and at ASI, there's two types of researchers that work in those companies. Well, in any pharmaceutical company, really. There's the medicinal chemists and there's the process chemists. So as I said, I worked in the process departments. So I'm familiar with biocatalysis because that was a trend that was starting when I was there. So what's the difference between the two? Medicinal chemistry, the chemists in that lab will always work on small scale. They will do reactions to discover a small amount of molecules, and they will test them they're at the forefront of research. So they are the ones who make like hundreds and thousands of chemicals together, and they take the compounds and they give them over to the biochemists or to the biologists that have all these more complex assays that can test whether the compound has the effect that they want or not. You know, does it treat migraine? Does it lower blood pressure? Does it combat diabetes? So we often don't know. We'd like to think that we do know, but we often don't know which compound is going to do the job. So we need an army of medicinal biochemists to generate hundreds of chemicals to see which one's going to work. Once they found the best compound, the compound that does the best job, that has the lowest, you need the smallest dose to have the largest effect, which is the ideal case, then they will take that one compound and it will be selected as a lead. So this compound will become the one that you will get on the market. So out of screening thousands of compounds, they found one compound that is like aspirin, and aspirin, that molecule will become the aspirin that you find in the bottle that you buy, or I guess that you get prescribed. Now, once you know which molecule you have, the goal is to create it on large scale because you'll have to supply the planet with it. So let's move this over. And this is where you have larger scale reactors. So these are classical reaction flasks. So you have a large glass flask that's able to withstand pressure, complex little tubings that will introduce different reagents. And you have the condensers, so you can reflux the reaction for those of you who've done chemistry. So the solvents come down and then you can distill off, and then you can transfer your reagent from one flask to the other. And here you have a gigantic flask that's being heated on a jacket that's going to be heated at 200 degrees, and then the whole reaction comes. So what's the point here? The point is let's move this over. So the point is, medicinal chemists will need to generate a lot of compounds. Once they find one, they give it to the process chemist, and now the process chemists their job will be to optimize the production of this one molecule that's been identified. This is where biocatalysis comes in. So if you can find a way to get bacteria or algae or microbes to replace all these tubings and all these reactors and all these reagents, then we can win as a society. So where do you start? So I'm going to take artemisinin as a case study for this class. So artemisinin, the structure is shown here. Do we care about the structure? No, it's just the idea. So artemisinin is a drug that is now, well, distributed widely around the planet, and it comes from this plant here, Artemisia. I don't remember the Latin name. So it's a common plant that is found about everywhere in the world, but the one strain of that plant that will produce this particular compound more than the others is actually found somewhere, I think, in the mountains in China. So now we can cultivate it on a large scale on the planet. Why are people interested in this particular drug? Well, because malaria. So it's one of the most effective anti-malarial drugs out there. So as you look at the whole world, all countries that are around the equator will have, and that are in very wet regions, will have a very high concentrations of mosquitoes. So I probably don't need to talk to you too much about how malaria gets spread. But long story short, it doesn't come from the mosquitoes. It's part of it. There's a small parasite that is in the blood of mammals, including humans, that when the mosquitoes will come in and suck the blood out of an infected person, when they go around and they will, let's say, sting another person, then they will actually carry and introduce the parasite into us, which will then cause malaria. So what I wanted to show here, so the amount of people that are affected by malaria on the planet is actually quite significant, because those, actually, if I zoom out, I'm not sure if it's by coincidence, but a lot of the countries that are affected by malaria also a lot of the countries that are the most populated. You have India, you have a lot of countries in Africa, you have Brazil. So all of these have very high populations. So when you look at the number of deaths that are caused by malaria, what you see, they have been increasing a lot, but there's been a significant decline after 2005, and now we're dropping. So there's two reasons for this. One is some technologies that are trying to control the reproduction cycles of mosquitoes, but the main one, because this is about the death, it's not about infections. So the main factor here is we have better drugs. So what is one of the main drugs that is the most effective is artemisinin. So there's a lot of research that has gone into making this available to, well, half the globe, basically. So you have to be able to produce artemisinin on a 3 billion dose scale on an annual basis if you want to be able to circumvent the growth or death of malaria. So this is such an important problem that I think the researchers that discovered artemisinin and studied its effect got the Nobel Prize, I think, it might be 2 years ago, 2018, I think. So for those of you interested in this particular story from a human and research point of view, you can look into the Nobel Prize. It was either in chemistry or it must have been in chemistry. So a bit of what I'm talking about today kind of relates tangentially to that story. Okay, so back to the question, if you need to have artemisinin and you have to create a lot of it on the planet, growing the plant will be too slow and it will be destructive because you have to destroy the plant to extract artemisinin from it. So the typical answer was to use traditional organic chemistry. So that is chemically synthesize it. So we would start from a simple molecule. I'm not going to take you through all the steps. And then the point is you get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, at least 15 steps to get to this. As I mentioned, anytime you do a chemical reaction, you drop the yield. You basically lose all along the way, you lose yield. So we start with a gigantic amount of starting material, and in the end, you end up with very little of this. So it's not very efficient. But more importantly is you have different reagents that use magnesium, that use toxic reagents, that use sulfurs, that use barium, that use zinc, that use boron, that use, here it's not shown, but Jones reoxidation uses chromium. So all these reagents, silicon, so all these reagents are, one, expensive, and a lot of them are metals, and most of them are toxic. So if you have to produce this on a half the planet scale, you generate a lot of waste. So this is what biocatalysis is trying to solve. So here's where, so I'm skipping ahead now into a completely different field. This got started by a lot of different scientists, but one of the persons that was the most active in the field and probably was the most successful was Jay Kiesling at Berkeley. So I'm going to come back to this in a second. The idea was, can we sequence, so when a plant creates artemisinin, it doesn't create artemisinin, from the same chemicals that chemists use. It uses things that are available in nature. So it starts from very simple components like pyruvic acid. Here, this is glyoxal triphosphate. Do we care about the names? No. The point is, these compounds, these chemicals are present in a plant in a natural state. They can be created from CO2, from the atmosphere. So this is carbon sequestration. So when a plant grows, it uses CO2 as the carbon source to create more of its structure. That's where these carbons, CH3, C, COOH, that's where they come from. So there's an extra layer of environmental friendliness that we can exploit with biocatalysis. Is the feedstocks come from the atmosphere, come from CO2, so it's kind of an extra way of using carbon sequestration. But I'm not going to talk about this today. So the point is, you get natural compounds that are present in a plant, and one enzyme, when assemble them, one enzyme will create this, one enzyme. So each of these arrows is a transformation carried out by an enzyme. So what a lot of scientists, including the ones that got the Nobel Prize, did is they identified which enzymes were responsible in the plant to put together the chemical components that eventually would lead to artemisinin. So for those of you who are who are paying attention, you are going to notice that actually the synthetic, like the natural biosynthesis of artemisinin in the plant includes way more steps than the chemical synthesis. But the plants are very efficient, and a lot of these chemicals are not going to be used only to create artemisinin. They're going to be used to create other components in the bacteria or the plant or the yeast. So what the scientists did is they identified each gene and they managed to clone the gene in an organism, and the Kiesling lab at Berkeley chose yeast. Why? Because it's a very well characterized organism. It's easy, like humans have a very long history of using it. So this is now the same kind of scheme that focuses only on the enzymes. So each of the compounds are being transformed by these enzymes, ERG13, ERG12, and then you have IDI1 and FPP, and then you have ERG9, and then you have cytochrome P71. Do you need to know this? No. The point is they've been identified. And if they've been identified, it means that now you know the gene. And if you know the gene, you can extract the gene and clone it into plasmids that we humans have now been pretty good at using artificially. So that's what the Kiesling lab did. This is probably 12 years ago. And they started, and the whole idea was, if you want to produce pure artemisinin, what you would need to do is to take a yeast strain that is well characterized, that they know, and introduce genes for amorphodiene production. So I'm not going to go over this. This is just one of the intermediates of the molecule. So we get 1, 2, 3 enzymes, genes that are going to create, that are being used to create 40 grams per liter of the compound that they want. And then they have another yeast strain that when it sees this particular compound will use 3 more genes to keep the synthesis going. And then I'm not going to go through every single step here. The point is you have all these arrows represent a gene, so an enzyme, who's transforming the original starting material into eventually this particular precursor. So this is artemisinic acid. So it's still not possible for us to be able to get pure artemisinin to be created by the bacteria. Why? Because artemisinin, because of this particular peroxide bridge here, it's a strange molecule, is what's killing, is the component that is killing the parasites. So this is the component of the molecule that is actually destroying living things. So you cannot have your microbes take it all the way there, because as they do this, once they create the final product, they kill themselves. So the whole synthesis, the whole is stopping at the last intermediate that is not biologically active. Now the chemists do one last step on it, and then they sell this part. So all of this is created by the microbes or yeast, and the last one is chemically modified. Okay, so moving on. So you kind of get the idea here. So I'm not going to belabor the point. You have a lot of different enzymes that have been identified. So for those of you interested in this, you can dive into it. Cloning is the key step in that process. So you have your bacteria or your yeast. I'm showing you bacteria here simply because it's the easy image I found, that carries its own DNA, that carries its own genes. But bacteria is very good at uptaking circular plasmids. And all of these plasmids, I'm showing a sequence here. They're not the ones that are being used for artemisinin, but you probably have seen this before. So all of these plasmids are DNA, circular DNA, double-stranded DNA that carry different components that are necessary for the expression of a gene, expression of a protein, and you can clone sequences of the gene that you want, that is these guys here. And if they're all present in the microbe, then the microbe is going to produce the enzymes, and as it sees some starting materials, that is going to start the whole cascade to create the desired product. So this is kind of the workflow that people do. Again, different angle, different image, but same idea, where now you have this whole envelope here is the double cell wall of your microbe, which would be a bacterium, an algae, or yeast, that carries now genes that were originally in a plant that has been now cloned into a different organism. So they will do their job. They will start from acetyl-CoA all the way up to, and after you get your final intermediate, which is gets secreted by the bacteria, or when the bacteria dies or the yeast dies, then the whole envelope breaks down and the compound that you wanted is getting released in the supernatant, and it can be extracted from there. So this whole field here is now called synthetic biology. So I started by saying this is biocatalysis, which technically it is. Each single step here is biocatalysis. But now synthetic biology is you're taking an organism, you're genetically modifying it to do something that it was never designed to do in the first place. So can it be scary? I don't think so. I don't think this is a big problem. Why? Because the organisms that could, let's say, escape. Let's go to the next slide here. This is how basically it's done now. So you have a gigantic reactor that is full of water with your algae or yeast. Feedstock will be like sugar or tryptone or whatever you need to keep the cells going. And you get some recirculation, and then you basically extract the compound you want. And that gets put into a truck and shipped somewhere. This is an example where you have biofuel. So where the compound, instead of being artemisinin that's being produced, is basically precursors for diesel or other types of fuels like gasoline. Even though it doesn't look like gasoline could be produced, this is actually how it got produced in the first place in the fossil, I don't want to say record, but in the fossil, deep into the ground after long-term fermentation. So I lost a little idea here that I want to bring in synthetic biology. I lost my idea. So hopefully you get the sense that as long as you get some feedstock that comes in, the genetic, oh yes, what if it escapes? Let's say this bacterium is, you get a leak. This particular pipe here gets leaked and all the bacteria actually leak out and spills out. Is this a problem for the environment? Probably not. Because why? The bacteria have been originally, or the yeast have been originally modified to express something that's not necessary to them. It's like they have been modified to use proteins and resources and energy and food to produce something that is not required for their survival at all. It's required for us because we want it, but it's not necessary to survival. Therefore, from an evolution standpoint, as soon as this bacterium leaks out, it's going to be at a disadvantage with all the other bacteria or other yeast that exist in nature right now. So that means if it leaks out, it's going to probably die of its natural death because everything else is going to compete better with it than itself. Or one way to look about this is the environment outside the reactor is not ideal for the bacteria. Therefore, it's not impossible that they might mutate, but it's highly unlikely. And if they did mutate to survive, well, you would just have a bacteria that would create artemisinic acid, which probably would not be a big issue for anyone. It's not like this is toxic to, well, if anything, it might be toxic to malaria, so maybe that's a good thing. But I'm not going to dive into too much of the chemical environment problems here. Okay, so to kind of finish off, so what does that mean? It means that now this field of synthetic biology is very well ingrained although invisible to most people because this is something that's boring and people don't like science. That's not true. But it's not something that's sexy to talk about. So you now have researchers that are working very hard to make, grams and ton scale of different chemicals. Sometimes they're pharmaceuticals, sometimes they're biofuel, sometimes they're fragrances. And this is just to show, this is a company, Enzymical Chemical Solutions. I think these guys are in California. This is in Zurich in Germany and in Switzerland, where this is now commonplace. You have these very big reactors. Although it looks like it's chemistry, most of it is all water and buffers and bacteria that are being carried over. So the visuals looks the same, but what's inside any of these tubings and reactors is different than traditional organic chemistry. And a lot of academic labs are pushing the frontier for this field. So there's a lot of different labs in Canada on every campus, I would say, that do this kind of research. So this is kind of a small reactor on a small scale for research that's being pushed, where all the tubings and the buffers are being pushed into your incubator. So this is what, you as a researcher, if you do a graduate degree in synthetic biology or biocatalysis, that's what you would be working from or with. So this is an image from one of the labs at Concordia University. Every single campus has people that works on this. I would presume they're either in chemistry departments or biochemistry departments. Here on our own campus, we have the Dan group in chemistry who works on synthetic biology to create anti-cancer, not to create, well, to get bacteria to create anti-cancer compounds. We have the Walther's lab who works in enzymology also, that is not exactly designing, that is not using synthetic biology, but is definitely studying enzymes to get the enzymes that are present in certain bacteria to do things that are not natural so that we can use them for larger reactions. So I'm going to end with this biocatalysis paper here. So this is basically the paper I talked about earlier. So if you're interested in this, the Nature reviews. These are examples of applications that are now very well established in terms of biocatalysis. So you get materials that can be created from biocatalysis. You get fine chemicals. So this is vanillin. So when you guys bake cookies and you put vanillin, your synthetic vanillin actually comes from, it's been produced. So vanilla is extracted from the plant, vanilla. So people have extracted the gene from vanilla and now get the vanillin to be produced in bacteria on large scale without damaging vanilla of the plant. So a lot of fragrances, so compounds that are complex, volatile, that smell good, are being produced this way. So on our campus, the Mahmoud Lab works on these types of projects, especially from lavender. In pharmaceuticals, this is examples of different drugs. So penicillin here is now produced not by chemical means, but mostly by biocatalysis now. This is the example I talked about. You have biopharmaceuticals, so those are antibodies. So as we're very well aware of now, antibodies are important. And also in food, so aspartame, not that I would recommend anyone to add aspartame to their food, but a lot of food components can now be produced instead of using traditional chemistry, can be produced using biocatalysis. And I put the names here just to show that or to remind myself that I should talk about Kate Ryan. So Katie Ryan here is a researcher at UBC Vancouver, who's, an author on that paper. So she works on biocatalysis also. Nick Turner is a researcher at Manchester University, who's probably, I don't want to say the father of the field, but is probably one of the most prominent enzymologists on the planet. And what they do is they take the sequences of a protein, modify them so it can do something that nature usually does not do by its own, which does not mean that it's deleterious to other sources of life. So the last thing I need to address is limitation. So is biocatalysis going to save the world? Well, I don't know. But there's things it cannot do. So you have to start from something that exists in nature. It is not possible for scientists now to get an enzyme and create it from scratch. So we have to take a gene that already exists and we can tweak it, but we cannot deviate too much. Second, the substrate must fit in the freaking enzyme. That means the enzyme cannot do all kinds of reactions on everything. The enzyme has been evolved to bind only one substrate. So what do I mean by this is the binding site of your enzyme is actually, nature has evolved it to fit only what it's supposed to react on, your substrate. Because otherwise your enzymes would be transforming everything in your cells all the time, and it's not happening. Each enzyme is doing only one thing on one substrate, and it basically looks at everything else in the cell and does not do anything with it. Why? Because the substrate doesn't fit in there. So that means if you want to get a transformation going, your substrate must be able to fit in the enzyme. And another aspect, well, I guess maybe one thing I should mention. No, I'll keep this for later. So, and another aspect is genetic engineering can be a long process. So I said you can genetically engineer your enzyme to do something that was not natural, or you can divert its chemistry. So you can take a substrate here. If you take an enzyme, you can genetically engineer it, so it's going to add a chloride here, or with another mutation, it's going to introduce a chloride here. But doing this is a very long cycle. It's hit and miss. Like at this stage, a lot of it is randomness. So you have to screen and screen and screen and screen, and eventually maybe you're going to hit something. So we're not there yet. And one last aspect is that biocatalysis is only good for process chemistry. What do I mean by this? Is, as I said in the beginning, the medicinal chemists generate hundreds of compounds, and once they find the one, they give it to the process chemist. So that means what they do is, typical job of a medicinal chemist is they're going to, let's say, try to take, this is an example with, let's say, morphine. So you have morphine, and it's addictive. So it's a good anti-pain compound, but it's addictive. So what the chemists are trying to do is to try to find if they can make modifications around the molecule to keep the anti-pain function, but remove the addictive component. So what they've done is they replace different functional groups by different other groups, and you see that the potency, so the activity, either decreases or increases. So this is a more active compound than morphine, but it's also addictive. So I'm going to cut to the chase here, and basically what they found is adding this particular side chain and these groups instead of these. So now the structure is a little different, actually quite different. You have a compound that's more potent, so 4 times more potent, more active than morphine in terms of pain, and now it's non-addictive. So this is the job of a medicinal chemist. An enzyme cannot do this. You cannot take an enzyme and say, I want to have this, I want to have this, I want to have this, because you don't know which one's going to work. But once you have found, so you have to use chemical tools to generate all these analogs of the compound you want. Once you found the one you want, then you give it to the process chemist. And it's their job now to find a way to spend all the time and energy and resources to make this one compound on large scale using biocatalysis. Why? Again, because genetic engineering is long and it's tedious. So you cannot afford to have, let's put this in the center. You cannot afford to spend all the time to create all analogs if you don't know which one's going to be the winning one. You're going to do all that energy only when you know this is going to be the compound you want to produce on massive scale. Okay, with that, I will terminate. I will stop the lecture. So we're at 1 hour now. So I hope I managed to convince you that industrial biocatalysis can provide a lot of solutions for the future of production of chemicals in a manner that is more efficient than traditional chemistry, and hopefully also environmentally friendly. Already by getting rid of solvents, you're helping, when I say solvents, I mean chemical solvents as opposed to water. You're helping chemical waste reduction. And also by using reagents that are by not having to use reagents that are very expensive, you're also decreasing the cost of making the compounds, whether it's a polymer, a material, fragrance, or any use you have. By decreasing the cost, you're also making it more accessible to more people in the world, not just the first world that is more rich. You can allow, if you can ship a strain of bacteria, a small vial that contains a few bacteria or a few yeast strain to anywhere in, let's say, in Africa or in India, or I guess, yeah, I'm not going to make a political claim here. Anywhere where they don't have great chemical industrial processes, they can just feed sugar in a beaker and stir the thing, and eventually the bacteria is going to grow, or the yeast is going to grow, and it's going to, as it dies, it's going to release the chemicals that you wanted. It can be extracted. So I guess one way to think about this is, instead of having access to complex chemical starting materials, the starting materials here are going to be either sugar or CO2. So with this, we'll stop the lecture here, and hopefully I managed to, well, give you a bit of more knowledge about a different field than food. And it's definitely an industrial aspect. And although we didn't spend much time talking about microbiology, microbiology is key because that's basically the reactor, the natural reactor for all these transformations. So on that note, stay strong and hopefully see you around on campus.